everybody. Welcome to our lecture for today, which is going to be focusing on gene mutation. Here's some of the learning outcome that is scheduled for our lecture today, including looking at some of the mutation, how those mutations may affect the gene expression, what type of proteins can we make, are these all of the mutations that we have are identified as harmful, or can we have mutations that are beneficial to us? And also, lastly, looking at, okay, if there is a mutation, what does your body do to try to fix the problem? So let's just start by looking at the nature of the gene variants. So there's a couple words that are going to be thrown out today, more specifically one of the words that are mutation. So the term mutation and polymorphisms have different or provide different information for the gene variation and inheritance nature of the gene. So when we talk about the word mutation, we're actually specifically talking about the DNA sequence where there is a change, and that change could affect a specific phenotype in that individual, but overall it's rare in a population. When we're talking about polymorphism, basically we're looking at a variation in a gene. So there's the changes that in the gene that are not as uncommon as a mutation. So mutations are relatively rare because of our ability to fix it, but these are less rare than a mutation, and they're going to be more obvious in the population. So we will see and talk about mutation a lot today because that's the base of our lecture, and basically looking at how these mutations may or may not affect us. So when we're talking about it in terms of mutation, we can hear the word recessive versus dominant. So recessive is basically any type of mutation that's going to cause us to lose the function, versus when it's dominant, it allows us to gain a function or gain of the function. The term mutant refers to the phenotype, which basically means the characteristics that are visible on the person. Get it be, let's say, eye color, hair color, or a change in a gene that led to you not being able to process a certain protein, accumulation of a certain protein in your body, etc. But it's an actual physical change that is that can be observed on the physical nature or on the body. In terms of mutation, mutation are a part of who we are. It is important for the concept of evolution. So as I mentioned before, not all the mutations are harmful. Some of the mutations can benefit us and help certain population to grow and develop under certain difficulties or challenges from the environment. But our evolutionary relatedness to other species also allow us to study many mutations that are not related to humans, but because of our relative closeness in the genetic information, it allows us to study it. Now, again, we're not going to focus on any other animals or any other organism. Our focus is going to be on the mutations that are visible in humans and how each type of mutation may affect the human either negatively or positively. Now, before we go into details about these types of mutation, I want you guys to understand that there are mutations that can happen at different levels. We can have mutation that affects the gametes. So basically the germ line that forms either the oocyte or the sperm are mutated. So this change happens at the DNA replication phases of meiosis, which is basically your interphase, if you guys remember. So this is happening in interphase 1, where the genetic information is did not replicate properly. And because it is at the meiosis level, it means it's going to be in the sperm and the oocyte. It basically can be passed on to the offspring of that generation, of the next generation. Somatic mutation, on the other hand, we're talking about cells that you already have. So this is associated with mitotic cell division. So this is going to affect a specific category of cells. So the DNA replication, again, it is still going to be in the interphase of the cell division process, but associated with mitosis. And therefore, it only affects the descendants of a certain group of cells. So a person with a somatic mutation have what we call somatic mosaicism, which basically, again, you will not see that throughout their whole body, rather than you only see it in parts or sections of their body. And I have a couple examples for you. One of them is associated with a mutation seen in the dogs. So when you have a genotype EE, which is basically means they're both recessive, you have, EE in the color gene of the dog, and then you typically going to see a golden or cream color animal, versus if you have either capital E lowercase or EE, which means both of the genes are dominant, your color is going to be on the darker, so black and chocolate. So this is if this is associated with the genotype. Now, if you have somatic mutation, again, it means some of the cells related to the skin color of this dog have changed, have undergone change. Not every cell, but certain group of cells. So that somatic mutation during the replication process of the DNA causes pigmentation patch, as you can see right here. So the rest of the dog has still their normal color, but you have patches of that, the other pigment, the capital E lowercase e being visualized because of the somatic mutation. Now, somatic mosaicism, again, you kind of have your original DNA, right? When somatic cells mutate in one individual, not all of the cells of that individual have the mutation. So again, kind of highlighting this. So these pink cells that you see right here are identified as normal, but because of this little mutation of G2A, some of the cells have been mutated. Now, because not all of the cells are affected by this mutation, again, this is considered a somatic mosaicism. So you can see that only parts of their body of this individual has developed those characteristics. So this is patterns, again, arising from affected cells that have migrated and developed in different regions of the body. Sometimes that migration is significantly less, where you don't see that mosaicism throughout the body, and it would be kind of localized to certain regions, and sometimes it would be more dramatic, where you can see it throughout their body. An example of the somatic mosaicism in a human is what we call the Porteous syndrome, which is relatively rare. It is inherited again. It is a genetic disease associated with mitotic cell division, again, because we're using the word somatic. It's not something that's earned from the parents during the process of fertilization. Now, what this condition does, it causes an excessive growth in the bones, skin, and some of the organs and tissues. So in this individual, what you notice is there is a significant abnormal growth of the bone and the skin in the hands, which causes that, again, that overgrown and abnormality. Because this is a somatic mosaicism, depending on which part of the body these cells are located, this abnormality would be asymmetrical. So you only part of the body will replace it, but not the other side of the body. And again, in this case, the parts of the body for this individual being affected is one side of the body is less abnormal, and the other side has grown more significantly in both the brachial or the arm region as well as the hand region. Now, mutations can alter proteins because, again, if you want to kind of go back to what we have talked about, DNA can be used to make copies, which are known as mRNA, and that is used to make your protein. So that's something kind of you have to already know, like basically the back of your hand when we talk about genetics. So it is important for us to identify how the mutations are basically causing the symptoms that can be clinically identified and characterized. And that's going to help us understand the mutation better and the importance of that gene or that protein within our body. Now, some of the examples of the mutation that does cause disease, and this is very limited, there are literally quite a few of those, hundreds of them, some very rare, some not so rare. But one of the more common one I think that people are aware of are associated with beta globin gene. This is beta globin gene. If you're interested, it's actually a gene in a protein called hemoglobin that is found inside your red blood cell for the purpose of carrying oxygen molecule throughout the body. So if there is an abnormality or gene mutation within the beta globin, it can cause a medical condition called sickle cell anemia, or it cause a problem called thalassemia. And I'll talk about that in just a few slides in a minute. Another mutation that can cause alteration or change in your protein is a collagen gene. Collagen is a very common protein that is found in many parts of your body, including your skin, your nails, and your hair. And some of these problems are associated with, some of these genetic conditions that are developed include Ehlers-Danlos syndrome, chondrodysplasia syndrome, as well as the Alport syndrome. Now, let's take a look at some of, these genetic mutation that's altering our protein. So we already mentioned sickle cell anemia, but let's dive into what is actually happening when somebody is diagnosed with sickle cell anemia. So right here, you have the normal hemoglobin on the first picture. So your sequence is CTC, which creates an mRNA, a strand GAG, which encodes for glycine. Now notice that this case, there is no aggregation within the hemoglobin proteins that are found inside my red blood cells. They are all separated, beautifully not connected to each other. They're not aggregating together. And therefore, the shape of cells that you create are the beautiful round cells that you have right here, these kind of concave looking cells. Now, if we replace the T in the middle with a letter A, so replacing a thymine with adenine, then your sequence for mRNA is changed to uracil, and now completely encodes for a different protein called valine. Now, because of the presence of valine, those hemoglobin proteins that are inside my red blood cells are now going to form aggregates. So instead of having them as these beautiful individual hemoglobin structures, they form these really large aggregates. And because there are so many hemoglobin and they form these really long extensions inside the cell, it causes literally the cell to bend to form these sickle cell shapes. And these cells are actually very dangerous. They can, not dangerous in terms of health, you can still survive them, but they can get lodged inside the narrow blood vessels, which are basically your capillaries, and they are going to cut off blood supplies to specific regions of the body. These blockages can cause significant pain, especially in the joints of the individual. And notice that these cells, because of their abnormal shape, they get marked for destruction at a higher rate. And therefore, your liver and spleen are going to destroy them. And when you destroy red blood cells in a really high abnormal rate, and then you have low red blood cell count, which is known as anemia. So this is anemia, basically means low RBC count. Now, another medical condition that we have is thalassemia. This is also caused by the protein hemoglobin. So there is a mutation in the beta chains of the hemoglobin. And instead of having a specific number of these chains form to make your beta globin, there are too few of those are formed. And I'm not going to go into detail. If you're looking at a hemoglobin protein as a whole, so this is what I'm drawing is a hemoglobin. You have 2 alpha chains and 2 beta chains. If you have thalassemia, you won't have as many beta chains, rather you have excess of alpha. So now you have, let's say, 3 alphas and 1 beta for every hemoglobin. So this excess in the alpha globin prevents the formation of the proper hemoglobin molecules because it's not what it's supposed to be. So because hemoglobin contains a significant amount of iron, because those are the carriers of your oxygen, these extra irons that are were to be utilized for hemoglobin synthesis are not being utilized, and therefore they accumulate inside the body. And the extra iron can cause damage to your liver, to your heart, to endocrine glands such as your pituitary gland, your thyroid gland, your suprarenal glands, which is also known as your adrenal gland, etc. Now, thalassemia, there are two cases, what we consider a minor case, which is where you have a dominant gene and a recessive gene combined together. So the effect of it is not as significant. But if you have thalassemia as a result of the homozygous mutations, then that's going to be a major thalassemia when, again, this is going to cause all of your red blood cells to develop the same symptoms, which is problematic for your red blood cells. The other type of mutation we talked about and protein alteration is modification into the collagen proteins. So the gene expression for collagen. Now, collagen is very important for us because it actually found in many of our connective tissue. If you guys recall, connective tissue is one of the four major tissue types we have in our body. It is present, as I mentioned before, in your teeth. It's present in your ligaments and tendons, so basically connecting muscles to bones, bones to bone. A good majority of your dermal portion of the skin is collagen. Your cartilage tissue and your bone tissue also contains significant amount of collagen. So most of the connective tissue have high levels of collagen. So there are 35 collagen genes that encode for more than 20 different types of collagen molecules. So when I use the word collagen, think about them as a category of proteins. There are minor variations between them, giving us the 20 different type of collagen. So for instance, collagen found in a ligament does not necessarily identical to the one found in the skin. Now, if you have mutations in the genes for collagen, you can cause major problems, right? Because it's found in quite a few of your tissues. Now, we're not going to touch upon all of the medical conditions. I want to kind of mention three main ones that I had it listed before. Ehlers-Danlos syndrome is one of the major ones, which I think it's kind of helped you guys visualize it. So you can see in the regular collagen protein, you have these ends. And these ends needs to be trimmed, basically cut off, right? You kind of like think about that as those cutters that come down on either side of the collagen and cut off the extra pieces. Now, if you do have the mutation in this case, you are preventing those ends to be cut off. So you will have the collagen as this long, much longer structure. So collagen molecule cannot assemble. And as a result, the skin is going to be super stretchy. So not a good thing to happen. The other two syndrome that I mentioned was chondrodysplasia, which is can be a result of either deletion, insertion, or missense mutation, which I'll talk about them in just a minute. But as far as you know that this is a mutation in the collagen fiber, I'm fine with that. But why is this such an important medical condition? Because it can affect the growth as well as cause deformation in the joints. So again, it's affecting more specifically the bones and the joints, collagen that are found in the bones and the joints. Alport syndrome, again, there are mutations, and these mutations are going to affect other tissues and cause major problem with the cartilage tissue in the ear, causing deafness and also cause inflammation of the kidneys because there are some collagen tissues associated with that. You can look at some of the other medical conditions like osteogenesis imperfecta, type 1, causes easily broken bones, blue eyes to be, the white of the eye to be more bluish, and also deafness. But if you look at the list, there is a significant effect on the major organs that I mentioned, which is tendons, bones, joints, ligaments, as well as problems with the ear. Now, we have talked about mutations. I have kind of thrown out some words about mutation, but let's take a look at what actually causes the mutations. Now, mutations can happen spontaneously because you basically happen to replicate that genetic information very often. So even though you have mechanisms that fix the problem, you still have so many of these replication happening that the chances are that there is going to be a problem. You're not going to be 100% accurate. So that's known as de novo or new mutations. This is not caused by exposure to anything. It just happens naturally. And again, in most cases, we do have mechanisms that can fix the problem. But more commonly, and especially in today's society, we actually get continuous exposures to various chemicals and radiation. And these agents that can basically cause these chemicals or the radiation that can cause mutation in our genetic informations are known as mutagens. And many of these mutagens will actually eventually lead to the development of cancers because of the abnormal growth of the cell caused by these mutagens. Therefore, they're known as carcinogens. So, mutations or mutagens, if those mutations lead to cancer, we refer to those mutating agents as carcinogens. Some of the examples of the carcinogens as are alkylating agents that help that removes a base. The X-ray, which is at high dosage, can cause breakdown of chromosomes. And UV radiation, which is unfortunately in Southern California, we expose ourselves significantly when you go sunbathing or going tanning, which causes what we call thymine dimers as one of the more common versions. And thymine dimers is literally, and I don't know if I have a picture of this, thymine dimers is basically if you have like this sequence, let's say A, T, T, A, and this is the copy sequence, you're supposed to have lines going this way, connecting these two sequences together. But in the case of the dimer, you're creating bonds within the same chain. So there is a bend that's created as a result of these two T's next to each other, forming and connecting a covalent bond with each other, which is again not ideal because it takes away from the original shape of the DNA. So we have understood two types of mutation or two causes of mutation, things that are considered mutagens or just a spontaneous change in DNA. Now, rate of spontaneous mutations, again, no connection to any type of mutagens, can be different depending on the gene. If the gene is undergoing mitosis at a significantly higher rate, the chance of mutation on those cells would be more common. Also, if you have a gene that, let's say, is 16,000 bases or 1,600 base long, has a higher chance of mutation versus a gene that's only, let's say, 500 base long, because you are replicating a longer sequence, therefore the chances are you may make more mistakes. Now, each individual has multiple new mutations. That's normal. But is the problem, again, as we go and expand on this, is this mutation going to cause major change in the gene expression and alter the protein in a way that can cause medical conditions? Now, a spontaneous mutation can also happen as the egg or the sperm is being developed in the sex organs. So, for instance, if you have a parent mutation, either a sperm or the oocyte within the reproductive organs, testes, and ovaries, that spontaneous mutation can basically get passed down to the next generation through the sex cells. And this type of spontaneous mutation is called gonadal mosaicism. And again, it's getting passed down because the genetic mutation or the problem arises from where the sperm and the egg was being developed inside the testes and ovaries. Another part of your cell that undergo mutation is the mitochondria. So Mitochondria is the only organelle inside your cell that has its own genetic information other than the nucleus. But even though it replicates its genetic information, it does not have the capacity to repair itself. Because of that lack of tools to fix the replication problems, it is more susceptible to mutation or spontaneous mutation to remain inside the mitochondrial DNA. Now, if you look at the sequences of your DNA, there are regions that are going to be more susceptible to mutation. And most of these regions are where you have a short repetitive sequence, like as described here, your sequence of A's or T's, depending on which sequence you're looking at. That repetitive sequence interferes with proteins and enzymes that are responsible for fixing any problem with the genetic information and basically delays or stops the repairing process. Now, these regions, these repetitive sequences are known as mutational hotspots. And again, there are chances that you will have a mutation in them and your body is unable to fix it properly because the repair enzymes are not, sorry, I should say that pairing prevents the enzyme from fixing the issue. Now we're gonna dive into the types of mutations that can happen on our body. And again, how does that affect our gene expression and development of medical condition, possibly development of medical condition. One of the first things before I even talk about that, I want you guys to understand that if you have a gene, you may have one gene having multiple or different type of mutations in different people, but they all cause the same medical condition. And I believe I have a picture of that. So I just want to show you this. Sorry, I'm going to kind of move forward and I come back. So different mutation in a gene can cause the same disease. So this is the picture I have. So you have a medical condition called familial hypercholesterolemia, which is affecting the receptors for you to bring cholesterol into the body. So the first one has a specific type of mutation called missense, where one amino acid is switched with a different amino acid. You have another example, which is non-stop. The protein is much shorter than what it's supposed to be. And then in this case, you have added 4 bases or 4 nucleotides to the sequence. So the sequence of the amino acid looks different in that region, I should say. Now, regardless of which of these mutations you have, the final medical condition is a receptor that is not functional and therefore causing the medical condition. Now, I'm going to backtrack. I apologize for that. Now, again, we talk about type of mutation. I'm not going to talk about this in this picture, even though this is a really, really nice picture for you guys to understand the analogy of the various type of mutation we can have. I would recommend after you visit the rest of the PowerPoint presentations, come back and see if you guys can understand how this mutation has happened. But we're going to talk about the mutation in terms of what they do and how they could affect the protein synthesis. So point mutation. So point mutation is basically, as the word point implies, it's a single change or mutation in one nucleotide. And this is, again, single DNA base being changed. So A, T, C, or G being changed to something else. Now, there are 2 different types of point mutation, what we call transitional or transition and what we call transversion. Now, if you guys recall, from when we talked about DNA structure, we mentioned that there are 2 types of structures that make our 4 bases. What we call purines, which have the larger structures, so one of these 2, or pyrimidines that has one of the 2 smaller structures. Now, purines are encode either for A or G, and pyrimidines encodes for C and T. So if you are doing a transitional point mutational or transition point mutation, you're simply switching one pyridine for another one pyrene for another pyrene. And for instance, A becomes G or G becomes A, or C becomes T or T becomes C. This is called transition. And if you notice, there are shown by the blue arrows that going back and forth. Versus transversion point mutation, you are converting a purine to a pyrimidine or a pyrimidine to a purine, which is represented by the red arrows. So G becoming a T or T becoming a G, A becoming a C, C becoming an A, or A becoming a T or T becoming an A, or vice versa for C and G. So the differences again in transition versus transversion is, are we keeping the same structures of purine or pyrimidine, or are we also, not other than changing the point mutation, we also changing the original structure from pyrimidine to purine or vice versa. Now, these point mutations that I talked about, let's look at the consequences. So one of the consequences of having these point mutations is called missense mutation, which causes a replacement of one amino acid with another amino acid. So you simply change the amino acid by adjusting or modifying that single individual nucleotide within your DNA sequence. Nonsense mutation is where you take a sequence and by changing that single point mutation, you change a regular amino acid to a stop codon, and therefore the protein basically ends. So you're producing a protein that's typically much shorter, and that proteins would be non-functional. Now, cells that do have these nonsense mutation, they typically mediate what we call nonsense-mediated decay, which basically takes that protein and break it down and get rid of it. Now, the stop codon that is changed to a coding sequence causes the lengthening of the protein. I believe I have a picture of this. So we're going to look at some of the vocabulary here. Now, notice here, there is some vocabulary that we're going to hear. Synonymous or silent simply means that there is no change that is causing there is a change, but that change is silent, as in nothing can be noted about the amino acid that would have originally been made versus the one that is made through this process of mutation. We also have non-synonymous, which basically means a mutation that has affected the amino acid sequence and therefore the protein. And then it has its own variation, which is what I'm going to talk about in just a minute. So let's look at the original sequence. The original sequence of the DNA is AAG, and the mRNA becomes AAG, and the protein that's being produced is lysine. Now, if I take the original DNA sequence and replace it with AAA instead of a G, so this is the one that's making the change, I will encode for AAA mRNA, and that is still a code for lysine. So even though there is a change in the DNA sequence and therefore mRNA sequence, there is no change in protein because both the mRNA AAG and AAA encode for lysine. Hence, we're using the word silent mutation because there is no change in the amino acid sequence. Now, non-synonymous, again, you will see and observe a change. One of these changes is called nonsense, which I just talked about. So instead of encoding for AAG, now you're encoding for TAG, which translates to UAG, and UAG is one of the 3 stop codons you have. So you literally stopped the making of that protein at that point. Now, this is called nonsense because the protein that's been made is not a complete protein. Sometimes the protein, let's say, is going to be a 400 amino acid protein, and nonsense happens at the position 50. So instead of being a 400 amino acid, you only have 50. Therefore, that protein is nonsense. It will not be able to make or work and do anything that it needs to do, and therefore it undergoes nonsense-mediated decay. The other type of non-synonymous mutation, point mutation that we have is a missense. So missense can be both conservative or non-conservative. What does that mean? So let's take the first example. So if you have a conservative missense point mutation, you are taking, let's say, AAG again and replacing the A with the letter G. Okay, so instead of saying AAG, now you have AGG, and your original amino acid from lysine is now has changed to arginine. So is this an actual change in the amino acid? And the answer is yes. However, if you look at the lysine, lysine is identified as a basic amino acid, and so does your arginine. So even though the sequence is changed, right? Even though amino acid is different, they're both still basic amino acid, which means this is considered conservative. You kept the nature of the amino acid, even though it's not the same amino acid. On the other hand, if you take the AAG original sequence and change it to ACG, now you're encoding for tyrimidine or THR, and this is a polar amino acid. So you're changing a basic amino acid to a polar amino acid. This is called non-conservative because the original nature of the amino acid was not saved. Now, a splice site mutation. So look at the word splice. So think about when we talked about creating your mature mRNA. We had a long sequence, which was made up of exons and introns. To go from an immature mRNA to a mature RNA, you were to remove the introns and connect the exons together. And when all the exons came together, you got your mature mRNA, which was used to encode for a protein. Now, a splice site mutation can happen two different ways. One type is what we call exon skipping. So instead of having, let's say, exon 1, 2, 3, and 4 being copied, so being connected together, one of these exons is skipped, and you end up with only 3 instead of 4 exons. The other one is when you keep an intron in instead of having it removed. So that can cause major problem with the protein because now you have an extra sequence that is in there that's going to be translated, which wasn't supposed to be part of the protein. So because your mRNA sequence is lengthened, the protein is also be lengthened. And because of those extra amino acids, you may not create a protein that's functional. Best example of it is cystic fibrosis mutation that is observed. And in terms of the exon skipping, we have a medical condition called familial dysautonomia that affects the development and survival of some of the nerve cells and is shorthand to FD. Now, let's demonstrate this to kind of a splice siding mutation on an image. Here is your sequence. You have the exon identified by letters and introns with numbers. Now, logically, if I was to make this protein properly, all of the introns would be removed, and I will end up with A, B, C, and D as my normal sequence. The first example you see right here, where it says protein lengthen, you are keeping one of the introns inside, right? So intron 2 does not get cut off. So you're going to have A and B, an insertion of the intron here, and then C and D. Now, because of that extra piece of intron in there, your protein is going to be longer and probably non-functional. On the other side, you have a skipping one of the exons. So instead of having A, B, C, and D, you're going to end up with A, B, D, because the exon C has been skipped over. So in this case, because one of them exons are missing the sequence of the amino acids, and therefore the protein would be shortened. Now, deletion and insertions. So this is the genetic code is being read in triplets, which we already know because of the codon nature of the amino acid and the process of translation. So anytime you have an mRNA, you will read it in a 3 nucleotide base or a codon. Now, nucleotides that are changing, not in multiples of 3s, so basically you're not changing one codon, can cause disruption in your reading frame. So that's referred to as a frame shift mutation. So if you're adding or deleting one of these if you're adding or deleting one of these individual nucleotide, your whole sequence of codon will be shifted, and therefore the amino acids that are being synthesized is nothing like the original one you were hoping to. So any deletion or insertion that happens not in a frame of 3 will cause a frame shift. So let's take a look. Let's look at this concept. Sorry, I'm going to go back because I thought I had a picture of this. I do actually have a picture. Sorry, I'm going to kind of, hopefully I don't give you a headache by going back and forth. So here is basically what I'm talking about. So frame shift right here. This is the example I told you to go back to. So the first 3 is the same. The second 3 is the same. And now instead of having BIG or big, I have in place a Q in there. So when you do a Q, now your 3 codon becomes QBIGFL. So instead of having words that does make sense to us, now we have words that have absolutely no sense to us. So that's exactly happened. So your amino acid sequence, if this was your original amino acid sequence, now has converted to amino acid code sequence that has absolutely no sense to us. But if we have deletion and insertion happening at 3s, you basically, in this case, you're missing 3 of them, which in this case was fly. And in the next example, you have insertion, which you inserted a brand new codon into there in a multiples of 3. So frame shift can happen at once, which causes major problem. If the amino acids are either added at 1 or 2 bases. It doesn't have to be 1. But if it happens on 3s, we call them either a deletion or insertion. Okay. So deletion and insertion examples that lead to major medical condition. For deletion, we have removal of the genetic information where there is literally part of the gene Y, which is what identifies sex of the baby as a boy, is deleted, and that causes male infertility, basically lack of ability to create offsprings. And an insertion adds the genetic material. This medical condition, one of the best examples, called Gaucher disease, where you have an insertion of a gene, insertion of a base, not a gene. This causes a major buildup of a specific type of fatty substances in some of the organs, such as your liver and spleen. And these built-up causes those organs to be enlarged. And can you imagine if you have so much fat in those tissues and they're enlarged, they're abnormal, and therefore they will not be able to carry out their function, which is very problematic when it comes to liver. Now, we have something called pseudogenes. And as the name implies, pseudogenes basically means fake genes, right? These are DNA sequences that are very similar to our actual genes, but they don't necessarily get translated, right? Hence the word pseudogene. They look like a gene, but they don't get a protein out of it. Now, many of these genes were actually at some point were a gene, and somehow these genes were duplicated, and mutation has occurred in them, which caused them to be pseudogenes. Now, in some medical conditions, what happens is the part of the pseudogene gets crossed over with your healthy, happy gene that encodes for a proper protein. And this disrupts, this kind of fluctuation causes an effect on the final protein synthesis. So again, kind of going back to that Gaucher disease that I mentioned earlier, is a result of crossover between a pseudogene and an actual gene. So an enzyme that's supposed to be produced called glucocerebrosidase is not being produced properly. So instead of having the gene being transcribed and translated properly, there is and I'll show you a picture of this because I think it helps. So here is the normal gene in black. Here's the pseudogene in kind of a striped colors. Where that crossover happens, right here between these regions 8 and 9. Now you have the original gene for the good part of it, and then the part of the pseudogene at the end of it. This is abnormal, which basically causes the medical condition called the Gaucher's disease. So it's very interesting to know that 90% of the sequence is similar, and that abnormality is actually located all the way kind of closer to the end of the sequence. So as you can see, is in the exons 8, 9, 10, and 11. Now, transposons. Transposons are basically known as jumping genes. These are the genes that go from one location to another. And if these genes happen, they can actually cause major mutations and cause major health issues and medical conditions. So one of these examples of these jumping genes that causes a frame shift that are not, again, the frame shift, if it's on the basis of 3, you may actually have a protein that's almost completely functional and normal. But if the frame shift happens because your frame shift is less than 3 or more than 3, and not in sequences of 3, multiples of 3 bases, that causes major health problems because those proteins are going to be non-functional. So an example in this case is hemophilia A, which is basically a medical condition that's associated with lack of blood clots. The transposon has jumped into a chromosome 22, which has genes that encode for clotting factors number 8. And when these clotting factors are absent, basically, or the protein is non-functional, you lose the ability to properly stop the bleeding. And to just make sure that you're clear, let me say if the transposon was like, let's say AAATTT, this transposon is okay because it has 3 and 3. Not okay, but again, because it's on the basis of 3, there is no, it's an insertion, and the chances are the protein would look pretty normal if the rest of the structures are not affected. But if your transposon, let's say it's AATTT, now you have a 3 codon, right? A 3 base codon, but a 2. So that's going to cause a frame shift. And that frame shift means that every amino acid after this point will be off. It will not be the same amino acid, and therefore can cause major mutational changes in the sequence of the mRNA, and therefore the protein being synthesized. Now, we did talk about silent mutation. Remember, silent mutation is where you basically have no change in the amino acid that being synthesized. For instance, if you go from CAA to CAG, both of these are encoding for a protein called glutamine, which means even though you do have A changing to G, which is a point mutation, you did not change the final amino acid that are being utilized, and therefore this is considered to be a silent mutation. Because the amino acid sequence didn't change, we also call that the synonymous codon, because again, encoding for either CAA or CAG did not change the sequence of the amino acid. Now, let's say we talked about all of these mutations. So it seems pretty horrendous because every time your DNA is replicated, you become more susceptible to these medical conditions. And that is true to some level. However, your body is also pretty good at making sure that these problems during the DNA replication, either through meiosis or mitosis, does get fixed. So we do have repairing mechanism inside our cells to make sure that the cells are working properly. Now, how do we do that? One of the first things that you want to note is that even though this mutation do take place, it's not as frequent, maybe, is 1 in 100 million of the base pairs. So it's very uncommon in the big scheme of things, yes, in a small scheme of things. But imagine, again, every cell is undergoing replication, then this number may not seem as big. Now, errors in the DNA replication or damage to the DNA creates those mutations. So again, either a spontaneous or damage to the DNA as a result of presence of mutagens can cause this. And if those mutagens lead to cancer, we call them carcinogens. Now, again, as I mentioned, can we fix the problem? Yes, we can repair them. Especially if you're on a younger age, the capacity to fix errors are significantly higher. But as we age, that capacity decreases. The type of repair depends on what type of damage or error we're looking at. So what are the types? We have what we call excision repair and what we call mismatch repair. So excision repair is when you have things like pyrimidine dimers or surrounding bases are observed within your DNA sequence. You go in, you cut off the structure, and you replace it up to 30 bases. And here's a picture of that, by the way, if you just want to kind of see it. So let's say UV rays comes in, it causes damage to your DNA sequence, and you create the thymine dimer, which is basically, again, a covalent bond forming between two T's. So the repair enzyme comes in. Here's your repair enzyme. The repair enzyme basically removes the incorrect sequences and replace it with the proper nucleotides. And then your DNA ligase comes into play and basically glue everything back into place. Mismatch repairs is when you have, let's say, a T is, instead of being a T, is an A, or a C is a G, etc. Here's a cool type. When you have that mismatch nucleotide base sequence happening, it actually creates a bulging inside the DNA sequence. So here is your original sequence, beautifully look, but then you have, instead of having an A here, you have a G, and that literally creates this little bulge inside the DNA sequence being replicated. So what does your body do? It actually brings an enzyme that cut off the incorrect or mismatched nucleotide base and replace it with the correct one. And then you have ligases that basically come in. Actually, no ligase is needed in this case. The correct sequence, the correct nucleotide comes in and replace the mismatched one, and the DNA replication continue to work. So again, do we have the capacity to do it? Yes. Do we catch a lot of mistakes? Yes. But what if we don't catch those mistakes? What we fail to catch those mistakes? So in this case, there is a protein that basically monitors and repairs the DNA, and this protein called P53. If this protein is damaged, this is the protein that basically monitors to make sure that the DNA is fixed itself. So if there is damage to that DNA, the cell goes like, nope, we gotta kill this cell because the chances are we're gonna create a lot of mistakes. Those mistakes cannot be fixed, and therefore that cell needs to be destroyed. Now, if you do have DNA repair disorders, which means these proteins that are responsible for coming and fixing the DNA are not present and your cell is not destroyed, then the mutation and the chromosomal break persists. So the problem with the chromosome and mutation persists and continues to happen. If these genes, repair genes, are not fixed, you become very susceptible to major mutations and problems in those cells. And it may lead to development of certain type of cancer if you have exposure to mutagens. So mutagen comes in, the cell gets affected, there are mutations, but because there is mutation in the repair genes already, then the problem created by the UV rays cannot be fixed. So the mutation remains. DNA repair disorders, again, cells that are basically unable to fix the mistakes of mutations, include a specific type of colon cancer called hereditary non-polyposis colon cancer. This is also known as Lynch syndrome. It in cases for 1 out of every 200 colon cancer that are observed, and if you don't know, colon cancer is relatively common and accounts for about 3% of it. This is a result of a problem, again, with the DNA repair protein that is supposed to fix the problem and itself is mutated. And therefore, the person becomes much more susceptible to specific type of cancers, including colon cancer, because those cells are damaged and therefore cannot replicate the DNA. So for this lecture, guys, I want to make sure you guys can distinguish the type of mutations. Focus on the understanding that if I give you a sequence and a mutated sequence, can you tell me what type of mutation is that going to be? Basically, can you go from a DNA sequence to an mRNA sequence to a translated sequence and tell me if the sequence is, let's say, is a nonsense, a frame shift, insertion, deletion, silent, etc. So categorizing the type of mutation, be able to briefly give me description of each of these processes. And if I just give you a normal sequence versus an abnormal sequence, can you tell me what exactly has happened to that sequence in terms of the identification of the mutations? That's about it. If you guys have any questions for me, please let me know. Otherwise, thank you and make sure to study.